Uh, very good afternoon to all the panelists and participants who have taken time off. And I'm pretty sure I was viewing the inaugural session. It was fantastic, and Mahesh Balgat had set a very nice stage of the entire workshop. And in the 30, 40 minutes that I have time, I would like to cover something very different, wherein I would leave the participant with some imagination as to what we can do with virus-like particle. That's going to be the future trend in vaccines, and there is a lot of promise in terms of research. So the disclaimer, name of any product or manufacturer used in the slide is only for the purpose of acknowledgement. It is neither a critique or nor review of their product process. Now, coming to the content, these are the content of my presentation. So let's start with viral vaccine manufacturing process. I'm pretty sure a lot of ground have been covered in biotherapeutic manufacturing during the workshop, and I saw quite talented lineup of speakers who were involved in delivering lectures. And I hope that I'm not repeating and preaching to the choir, but I just wanted to highlight something. And I just borrowed this particular slide from Cytiva website, which says virus vaccine manufacturing process. The reason I'm putting this is to highlight two things. One is, of course, there is automation and there's possibility of large-scale bioreactor-based manufacturing for virus vaccine. However, I hope people don't walk away from this workshop with an oversimplified view of what a viral vaccine production looks like. First of all, this particular virus vaccine, which is there, since I was not part of the entire session, speaks of a bioreactor production based on a vero cell. So there are cell-specific virus production cycle. So you start infecting the cell through a manufacturing seed trail. You undergo chromatography and purification, and sometimes you may have to freeze dry depending on if this is a lyophilized vaccine. And final filtration is what is mentioned. So there are two things. One is, I hope you realize people are getting used to looking at every virus like a coronavirus. It's not the flavivirus actually looks like this. It doesn't look like what was presented. And the other fact is, many viral vaccine production is not amenable for bioreactor manufacturing, and neither can they be filtered. So I'll touch upon some of these issues as we go along. And let me come straight away to why vaccine manufacturing for viral vaccine cannot be done in bioreactor all the time. Some of the viruses actually require adherent cell for cultivation, and that requires a fixed surface. And that the production scale would be something like a CS40 or CS52, depending on which manufacturer you're looking at. But it starts with a research grade, a CS2 or CS1 also. The CS refers to cell stack. And there is a static involvement, I mean, growth of cells. So there are issues when you do this kind of a production. The issue is not all viruses are adapted to a suspension culture or a vero cell culture. So like, for instance, hepatitis A is adapted to MRC5, and that's an adherent cell. It's a lung fibroblast and requires a surface to attach. And the moment you have a surface which is involved in attachment, there are scale-up issues, whereas this, although this picture looks small, but trust me, this is about 4.5 to 5 feet tall. So if you are producing something in you're handling live viruses in this kind of a container, you are exposing yourself to a huge risk. So there is a difficulty in manual intervention. Plus, Unlike a fermenter, there are not many in-process sampling that can happen. And filtration is not possible for most of the enveloped viruses, because the moment you start filtering, they would lose viability or even lose structure. So with that overview, I would probably jump to the immune response of virus. And I'm building a case why a VLP kind of a technology is very interesting and futuristic to look at. So the immune response to a virus is, this is a textbook knowledge, that you have a virus which is invading the cell, and once the infection happens, you have cytokines which alert the system, the MHC1 expression for the intracellular antigen presentation is overexpressed, and simultaneously the innate arm would recruit natural killer cell, an antigen presenting cell, which will be destroying the cell and phagocytosing that, then removing that. In the adaptive arm, because MHC1 is upregulated, T cells get involved, and if not, there are some B cells that can directly interact with viruses and present antigen to T cells using MHC class 2 presentation. That's all textbook basic. What is important is the immune response is also dependent on the antigen characteristics. The particulate antigens are the most potent antigen, and presence of pathogen-associated molecular patterns. These are present in polysaccharide, nucleic acid, repetitive structures are all part of pathogen-associated molecular patterns, and they all go on to stimulate the TLR. They are also called as TLR agonist, and in fact, some of these are also being used as adjuvant, as you would know. In coronavirus vaccines, a lot of manufacturers use CpG. CpG is nothing but a TLR agonist and can potentiate the immune response. And also, epitope density, how closely the epitopes are present, is also a characteristic that would enhance the immune response. And this is a research which is way back in 1974, that they found that if you have a chemical hapten, which is I forget the name, but it's a simple chemical. But if it is spaced within 5 to 10 nanometer, if you have 20 different of these chemicals, the chemicals themselves became immunogenic. So the epitope density is also something which drives the immune response. Now, with that in background, let's look at what the virus structure is. So a virus comprises, these are the terminologies that I would be using. A subunit is nothing but a polypeptide, and here you have human papillomavirus L1 protein. So L1 alone can self-assemble. So L1 expresses and assembles as a pentamer. So a protomer consists of a multiple composition assembly of a polypeptide that becomes protomer or capsomer, and multiple of these capsomer assemble as a virus particle. In this case, they have only shown L1. Actually, for HPV, there will be another capsid protein, which is L2, but it also takes DNA and packages into a virus structure. A capsid is essentially assembly of subunits or protomers, which basically is required to guard the nucleic acid of the virus or the genome of the virus. An envelope is derived from a host, which is membranous structure, lipid membrane, and sometimes a nucleic acid is also involved. When a membranous structure is present, then the genome is protected by a wrapped around protein structure, which is called nucleic acid, and they have a very discrete structure. In addition, almost all viruses, non-enveloped viruses, display an icosahedral symmetry, although even flavivirus display icosahedral symmetry, which is an enveloped virus. But that's a different discussion. I can get that into later. But a triangulation number T1 basically refers to same subunit repeated 60 times over. A triangulation, higher the triangulation number, more the subunit. But almost all of them display an icosahedral symmetry, which is 12 phases. Icosa is a 12-phase symmetry. And the larger the virus, the more the number of triangulation, the more number of subunits get involved. So it will be easier to assemble T1 symmetry, or sometimes it's up to T3 symmetry. But the moment you get into larger symmetries of up to T13 or T14, there are viruses which are that large. It is extremely difficult to assemble them, so they will be very, very slow. Now, what are the thermodynamic properties of the virus-like particle? We have to understand a virus has to infect, assemble quickly, come out and infect again. So it has to utilize something which is very, very simple, something like a Lego block, which is available to be assembled or disassembled repeatedly. So as a result, many viruses actually do not attain a thermodynamic minimum free energy to become stable, meaning they're always in transition of whether to stay in assembled state or disassembled state quickly. Virus, as I said, viruses have to synthesize rapidly, and because the infection cycle is fast, and the quick assembly and disassembly is also favored by the fact that there are no covalent linkages between the subunits. And a mere potential energy is enough to disassemble, which is more like a trigger. So a virus is potentially waiting to explode the moment something it finds its own receptor. So in fact, sometimes it can cause that much of a damage. We are already witnessing coronavirus does that. So in nutshell, a virus-like particle is in a thermodynamically metastable state. So these are all very, very critical information when we get into utilizing this technology for different uses. Now, let's get on to what is a virus-like particle. A virus-like particle is very similar to, as a name says, it is virus-like particle, which meaning here you have a viral particle and you have a virus-like particle. The difference is a virus-like particle has almost every protein similar to the virus particle. All the major proteins will be similar. It does not contain the genome, neither the membrane if it is membranous. So immunologically, from the external structure, it looks very, very similar to the virus, to the native virus. They are not infectious. They do not contain any of the genetic material and can be synthesized in vitro because, as I was detailing earlier, the virus structure is a repeated assembly of a subunit. So if you can produce a subunit, you could assemble a virus. Now, that assembly of a protein is actually in a heterologous system is the key to virus-like particle as a technology. So why do we do a VLP and why not grow the virus itself? First of all, the immune response mounted by a VLP and a virus are very, very similar. Not every virus can be cultured in vitro or also in an industrial scale. And the example for that is human papillomavirus. It grows, it infects the keratinocyte layer. There are no keratinocyte industrial scale culture model, although there are certain graft, immunologic graft model available wherein you have a limited scale of human papillomavirus that can be cultured in vitro, but that's not an industrial scale. So if you have to produce a virus in an industrial scale, you require alternative method. Plus, as I said, a VLP both is a particulate antigen and has a very high epitope density. I just want you to concentrate on some of these, you assume any of one of these color being an epitope, and because they are spaced very closely, just to give you a dimension, one strand of a alpha helical protein is about 4 nanometer. And if these are all beta sheets, you can imagine in a very, very close space, there are multiple epitopes that are being arranged, which is presented to immune system. And it does not require any biocontainment because there is no genetic material. It is not infectious. So producing a virus-like particle will be very, very easy. It is scalable and there's a possibility of even doing process automation. Back in Germany, I know that there is a particular lab that is producing in a robotic system the human papillomavirus pseudoviruses, which is completely robotic. From the time of infection to purification, it is going on automatically. So VLP technology is amenable for automation. However, as I said, the higher T number, the more the number of subunits that are involved in a virus-like particle or a virus structure, converting that into a virus-like particle will be very, very difficult for the simple reason that sometimes the stoichiometry of each of these subunits will be very difficult to maintain. And given we are looking at a spontaneous assembly of a virus particle, uh, uh, maintaining that stoichiometry and allowing individual components to come in an exact fashion is a huge challenge. Therefore, any virus that has a higher T number, it will be very difficult to convert that into a VLP. And as I said, VLP is a very, very safe way of manufacturing. It's a safe manufacturing alternative. So what is the example of a VLP vaccine? One is, of course, you have both human papillomavirus and hepatitis B, both are successful VLP vaccine. Let me take you through what is a human papillomavirus consisting of. It consists of a major capsid protein L1 and a minor capsid L2. It has a double-stranded DNA, which is 8 kilobases. And this is a very important feature. I'll come back to this in a while. So if you see, this is a picture I had shown earlier as well. You have L1 monomer, which forms a pentamer, which is capsomere, and it can assemble as a virus-like particle. However, if you have an L2, so you notice the difference between the VLP and authentic virus. The L2 basically sits inside the pentameric structure. So it binds all interacts with all 5 L1 and also binds the genomic DNA. And therefore, this is far more stable and compared to a virus-like particle. Yeah. So the very important feature here is if you have a virus-like particle consisting of L1 alone in HPV, in absence of the L2 monomer also, it can encapsulate the 8 kilobase genome. It does not recognize whether this is a HPV genome or a simple 8 kilobase plasmid, which is an interesting feature, which means you can use this as a delivery vehicle as well. Now, this particular vaccine has been used, and we know that it is sold globally as a prophylactic vaccine for cervical cancer. So when it comes to application of VLP, we have epitope display. What I mean by that is, let's assume this is an authentic L1 configuration, and these are the loops, which means these loops may or may not interact. Just in case if they are not interacting with the neighboring L1 capsid, then it is very much possible you could replace these loops with an immunodominant epitope from any other pathogen, and those can be displayed in multiple copies. Now, the advantage of that is you not only are displaying an immunodominant epitope in a virus-like particle, but you're also increasing the epitope density in a very close area. Plus, it becomes a particulate antigen, which means if you have an epitope display over a VLP, the possibility that it will have a higher immune response is very high. And as I said earlier, that this 8 kilobase genome is already, although it is part of an authentic virus, L1 can also package a 8 kilobase genome. So in the assembly, while all this assembly happens inside the cell intracellular, not only can this package the genome inside as a delivery vehicle, if you there are ways to disassemble the L1 particle, which is basically these L1 assembly happens because of interchain disulfide bonds. So if you have a reducing environment, the L1 will fall apart. So assume that you have a VLP, you keep that VLP in an environment which can encapsulate drug. So you basically encapsulate drug inside the virus-like particle, and these virus-like particles have a very specific receptor in some epithelial tissues. So in specific epithelial tissues, you could target drug delivery. So that's another mechanism by which you could utilize VLP technology. And last but not least, quality control assays can also be performed. In fact, a lot of coronavirus quality control assays are being performed by pseudovirus, which is nothing but a virus-like particle containing a pseudogenome, a reporter genome. So a VLP can also be utilized for creating a quality control assay. So in addition to the viruses per se, there are proteins that have the propensity to come together and form a self-assembled macromolecular structure. In this case, this is ferritin. Ferritin is very, very common across the biological kingdom, right from bacteria to human. We have different versions of ferritin. Humans have 3 different kinds. Bacteria have, E. coli has, Helicobacter has. This particular is a Helicobacter ferritin, in which the N-terminal is actually free, and the N-terminal does not interact with with any other subunit. So what these people have done is they have cloned as a genetic fusion using a linker so that it doesn't really destabilize the particle. Using a linker in the N-terminal fusion, you have HA of an influenza virus. And when they are expressed and assembled, you can see now this HA is being displayed into multiple copies. The advantage here is influenza virus per se is a membranous virus. It will be difficult to grow and probably have large enough titer. Whereas if you have ferritin, ferritin can be grown in E. coli, it can be grown in Pichia. So you are encountering a high cell density situation. And look at these electron micrographs, the epitope density that will be presented to the immune system will be very high. So the applications, a virus-like particle is just a start, but application for a self-assembling particle is actually huge. Virus-like particle happens to be one of the self-assembling particle. So in nature, there are several of these self-assembling molecules available, which could be utilized for displaying epitopes for various reasons, especially for creating vaccines. Now, to summarize, the manufacturing of vaccine in bioreactor is always not very straightforward. And I hope that is not the message that is going across, that a bioreactor is good for all biotherapeutic manufacturing. So, and the viruses mount very strong immune response because of very high epitope density per nanometer. And virus use simple mechanism to produce capsid, which means that if you have a lower T number virus-like particle or viruses, that uses probably only one subunit, which is repeated over several times. So producing these kind of a virus-like particles will be very, very straightforward. And VLPs are similar in terms of its immunogenicity as compared to the virus, and they are very, very safe to manufacture and can be scaled up very easily. So with that, I wanted to keep it very crisp and would like to have any question that is coming up for interaction.